Okay, and the last couple questions on here. What is the particular target organ for fever-suppressing drugs? You can answer that one. And this last one also takes just a minute to explain. Patients with a history of tuberculosis often show scars in the lungs and experience recurrent infection. Account for these effects on the basis of the inflammatory response. So to think about this, again, you kind of have to go back to thinking about what a scar looks like. If you have one anywhere nearby, take a look at it and you'll notice that it is much thicker than the other skin around it, right? It is a reorganization of fibrous tissue and always results in a lowered amount of circulatory tissue. So your circulation is lowered at those areas. So if you have a lowered amount of circulation heading or influxing that scar tissue, how do you think that would impact the ability for the body to sense and respond with the inflammatory response? Now, this is also true of people who are diabetic, long-term diabetics. If you've heard of these the symptoms before, many individuals who've had diabetes for a very long time can have diabetic ulcerations on their skin, on their hands and their feet. And the same concept is at play. I know it's not the lungs, but the same concept that it's a loss of circulation. So people who have been diabetic for a long period of time have a loss of circulation, and their blood pressure goes down, and they have a hard time getting blood pumped out to the extremities. And the same concept is at play. So think about why they might be more prone to develop continuous or reoccurring infections in their hands and their feet if they have a lowered circulation, and then answer that in terms of the inflammatory response. Okay, we're almost done. We have two more things to talk about here. This is interferon and complement system. So with these ones, I'll keep it as simple as possible. These are both a little bit complex, but we're gonna just look at one aspect of them. Interferons are small proteins produced by white blood cells and by tissue. They are antiviral. They can be produced in response to viruses, RNA, or some immune products. We're going to focus on antiviral interferons. And it works like this. So here's a virus infection. This is a mammalian cell, like one of any of your cells, like your liver cell. If a virus infects that, we remember the replication process is that virus comes in, it has to undergo uncoating, where that capsid comes off essentially, and what you're left with is viral nucleic acid. This is then replicated and synthesized into new virion particles, right, or virion proteins, and then you have spontaneous assembly and release. Now, during this process for animal cells, we have this evolutionarily adapted to these viral invaders, and one of the immediate responses to an infected cell, so as soon as your cell binds to that virus and that virus enters in and begins to uncoat, we have a signal that's propagated to our nucleus that changes the gene expression of the cell to produce these interferons, interferon gamma or others. This is again a small molecule. It's part of those cell-to-cell communication molecules that's sent out of an infected cell. This guy's definitely going to die, but before he does so, he's sending the alarm to all of his neighbors. And that interferon is released out into the environment, will bind to any neighboring cell, as every cell has these interferon receptors. And when this specific receptor ligand interaction occurs, that immediately sends a signal to this neighboring cell that is not infected to secrete or to begin producing antiviral proteins. And the strategy there is that once the cell receives that viral invader, which will happen since this virus is going to propagate and come and bind here, as soon as that virus enters and uncoats, releasing its nucleic acid, these antiviral proteins will immediately degrade the viral nucleic acid, which blocks viral replication and essentially stops the infection in its track. This is a really cool strategy our immune system has developed, and one that researchers, or actually clinicians, take advantage of to treat chronic infections like hepatitis. So forever, hepatitis was uncurable. This is an infection of the liver and always leads to, you can take viral suppressants, but if untreated, it leads to liver cirrhosis and often liver cancer, liver failure, and oftentimes death over time. So this new treatment, and I'm kind of going to answer one of your discussion questions, but you can genetically engineer mass production of this particular interferon itself, not the gene, but the protein, right? So the gene is cloned in, makes a whole bunch of interferon protein itself, and you can administer this interferon protein to someone who has a chronic infection. And the way that it would work and the way that it is successful in curing a chronic or an uncurable disease is, imagine this is your liver cell, and this is your entire liver. It gets infected with this bacteria or with this virus, and these guys are chronically infected. It doesn't go away. And that virus continues to propagate by continuing productive infections and infecting any new cell that is synthesized, right? Because you have continuous stem cell differentiation into liver hepatocytes. So normally you would continuously propagate that viral infection because any new cell is immediately infected by that virus and becomes infected, right? In this case, if you are taking an interferon treatment, and hepatitis does downregulate that interferon in the first place, but if you provide interferon treatment, then any new cell that's made by the liver, and your liver regenerates about every 7 years, so it is continuously synthesizing new hepatocytes, and every new hepatocyte would be immediately receiving that interferon signal, producing antiviral proteins, and would therefore be immune to that chronic viral infection. So this would be your new cell, and it's immune, and it will never get infected with that virus because it will continuously synthesize those proteins. And now every other cell that's made in that liver, all the new cells will also do the same thing, receive the interferon treatment and produce antiviral proteins. So essentially every newly generated hepatocyte will be immune to that viral infection. So over time, these infected cells will naturally die due to damage, but also due to the natural process of apoptosis. And what you would be left with are these new hepatocytes that have immunity to the virus. And after about 7 to 10 years on this interferon gamma treatment, then you have a new liver that has no viruses left in it because none of those cells are infected and they're protected too. It's a pretty amazing treatment. You can watch this video from our Canvas page. It shows you the same process there. And I just kind of answered this. How would you use genetic engineering to help stimulate an immune response that protects against viruses? You can re-answer that yourself, just make sure you're understanding that concept. And then this last component here is the complement system. Complement system is an extremely complicated system that we have as part of our innate immune defense system. If you've heard of C-reactive protein, what you're measuring is a component of the complement system. We're going to keep it kind of simple. This is a complex multi-duty system. It involves these basic proteins called complement proteins that are constantly in circulation and in tissue, and their job is to become activated and have very substantial responses as soon as they are stimulated. And we're actually going to switch this next slide to help understand this process. So these are the 3 ways. What you need to know are 3 ways that the complement system can become activated or initiated, and then the 3 results of an activated complement system. So again, complement proteins are just these proteins that are regularly synthesized and they're just released in circulation. So they're part of that surveillance network, but they're just proteins. But they're highly reactive proteins, and they're reactive. They're susceptible to proteolysis, which means lysis or breaking apart. As soon as this protein, the C3 protein or C5 protein, as soon as this is initiated or it binds to something that stimulates it, it will cleave itself into 2 subcomponents, C3a and C3b, that actually begin to function. So C3a has a significant function on, is a vasodilator, so part of that pro-inflammatory cytokine response that helps bring those white blood cells into sites of injured tissue. C3b, this fragment here, can go and coat bacteria, viruses, all of their surface proteins, and make them more susceptible to phagocytosis. This is a process called opsonization. Opsonization means coating to induce phagocytosis. So our macrophages have receptors for the C3b protein. So as soon as this is made, it can bind to these C3b proteins, but it's also super attracted to bacteria and viruses. So it'll coat bacteria and viruses, and as soon as it does, it makes them really, really easy to digest. This also can trigger further cascade reaction to initiate a signal propagation with other complement proteins like this. C5 can split and form C5a and C5b. This guy is also involved in the pro-inflammatory cytokine response. And then this guy can can join with other complement proteins, insert themselves in the membrane of gram-negative bacteria and cause the lysis of that cell. This is called the membrane attack complex. That last slide had a picture of these perforations that you physically can see under the microscope of a bacteria that's about to just die completely of lysis because they have no more membrane integrity. But I will say, as attractive and sort of sexy as this membrane attack complex is, sadly I have to report that it seems to be only effective against one gram-negative pathogen, which is Neisseria. So immunologists don't focus on the membrane attack complex very much, even though microbiologists think it's a pretty cool thing. So the ways that this complement protein can get activated are threefold. One is the binding directly to the bacterial surface proteins. So if this guy bumps into a bacterial protein, it can bind and begin to do these things here, initiate this cascade response. This process here is called the lectin pathway. Just think about this as a fungal organism. So fungi have large amounts of mannose proteins or mannose receptors, which are a type of sugar, and that can initiate a binding of this complement protein and again trigger this cascade reaction. And then the last one is antigen-antibody complexes. This means if there's an antigen, a piece of a bacteria from phagocytosis that is also bound to an antibody that's in circulation, then this antigen-antibody complex can initiate this cascade response. Alternately, if an antibody binds to even just a bacterial, virus, or toxin surface themselves, it doesn't have to be a phagocytosed thing, it can be a whole cell or a whole particle, whatever it is, that antigen-antibody complex can trigger this cascade reaction to generate these downstream effects. So you can answer these questions. How is complement involved in the inflammatory response, and how is it involved in phagocytosis? Those are really the only major things I want you to take away from the complement response. And that's it. Made it all the way through chapter 12, and I'll be back to talk to you about adaptive immune system next week.